Pretty eyes, pretty thighs, shawty, she a dime. Demon girl, evil eyes, she be telling lies. Slim waist.
So now the main reason why we're here is to learn about Xenobots. So my question is, what are Xenobots?
I think Mike should have a first crack at that one.
Well, yeah, I'll tell you what they are in the obvious sense, and then we can talk about what they really are, which that remains to be sort of determined. The simplest thing is just to talk about how they get here. So, what we are interested in, among many other things, is understanding how collections of cells make decisions about what they're going to do. Mm-hmm. And you would think that by looking at an embryo, which is very reliable, so frog eggs make frogs and acorns make oak trees, and so you would think that that process is very reliable and it's very hardwired, meaning that it's going to go the same way. And we're told about the genome and how the genome specifies your body, and so there's this idea that once you know the genome, you know what it's going to do. So we're interested in probing that paradigm and asking actually how do collections of smaller subunits, be they cells or robots in a swarm robotics context, how do larger scale kinds of systems scale up from components? So we did the following experiment. When I say we, there are kind of four main people on the team. So it's Josh, it's myself, it's Doug Blackiston, who's a staff scientist in my group. He's a biologist, and then Sam Kriegman, who's a computer scientist that was a student with Josh and then a postdoc briefly in my lab, and now he's got his own lab at Northwestern. So that's the team that I'm talking about. So what we wanted to do was we wanted to first of all find out how much plasticity standard cells have. So no genetic modification, no weird nanomaterials, just how much plasticity do they have on their own? And then, and Josh will talk about the computer science aspect of it. Can we use AI to really understand what it is that they're doing? Mm-hmm. So we basically, what we had done is to remove some of these skin cells from the early frog embryo, dissociate them, and then place them in a little kind of like a little hole, basically, where they would be in this protected environment. So this is a weird kind of engineering by subtraction, because we didn't add anything. We didn't give them new genes. We didn't use any weird chemicals. But what we did was we subtracted away most of the other cells that normally tell these cells what to do during embryogenesis. What do they tell them to do? They tell them to be a kind of a boring two-dimensional covering on the outside of the animal, keeping out the bacteria. So we said, okay, if we were liberated from that context, what else might you do if you could sort of reboot your multicellularity? And there's many things that they could have done. So the cells could have died. They could have crawled away from each other. They could have made a kind of a flat monolayer like cell culture. They could have done nothing. What instead they did was to get together into initially kind of a spherical little construct. And remember, this is just skin. So there's no nerves there. There's no other tissues. This is just. And what they do is frog skin has these little hairs called cilia, which are normally used. They kind of spin, and they're used to flow mucus down the body of the frog, right, to keep the pathogens sort of flowing off. So what they're able to do is use those little hairs to row against the water and to move. So they have spontaneous motion. So they move in various ways. They can go in circles. They can go straight. They can rotate in place. They can do a variety of things. And it became very clear right away that this was not only telling us some very interesting things about evolution and cell biology, but it's also a biorobotics platform. So we call these things Xenobots. Xenopus laevis is the name of the frog from which these cells come, and so they're called Xenobots. And then, we can talk about a variety of amazing behaviors, and then Josh will tell you what the computer science part of it is.
So I think to try and answer your question, the Xenobots remind me of the old story of the blind scientists, where one of them is touching the trunk of an elephant and the other's touching a leg and the other's touching a tail. So a lot of people, far beyond Mike and I, see different things when they look at the Xenobots. So you asked a great question. We could spend the rest of the time, I think, giving an answer to what a Xenobot is. So from my perspective, or the perspective of my lab, we're looking at Xenobots, or the living materials that Mike's lab is able to manipulate now, as a brand new set of Lego bricks, a brand new set of components from which we can build machines. And when I say we build machines, I mean either humans, so we could all sit down and try and come up with a design for a Xenobot. We're going to try and move these tissues around. We're going to liberate cells from the developmental trajectory in the way that Mike was mentioning and try and guess what's going to happen. To me, one of the exciting things about the Xenobots project is that it wasn't us that figured out how to rearrange the tissue. It's an AI program running on a supercomputer here in Vermont. So we have a lot of different names for Xenobots. One of them was computer designed organisms, to make clear that one of the things that's interesting about the Xenobots and many, many other things that are going to come in the coming years, they are being designed by an AI. They did not evolve here on Earth. They're not genetically modified by a human being who comes up with an idea about how to modify the genes. It was something dreamed up by an AI, and at the moment, built by a person, Doug Blackiston and Mike's crew. So I think that from a robotics and AI perspective, there's the implications of that are